My research project studied the quality of sushi-grade fish regarding the microbiology of the meat sample. The main question that this study aimed to answer was, is sushi-grade fish safer to eat raw? And that means safer than other ways that fish is prepared, such as buying it fresh or buying it frozen. So what exactly is sushi-grade fish? Sushi-grade fish is raw fish that is available for just the average consumer to purchase at the grocery store. And it is prepared properly for use in sushi, so it has been prepared following all the principles and rules that the FDA has put out to regulate this. And it is supposed to contain less bacteria than other types of fish, so it's safer to eat. Microbiological evolution of fish. So there are many types of bacteria found in fish meat. It actually has one of the highest bacteria counts of any type of meat because it has such a high water saturation, so it is great for bacteria growth. Some of the types of bacteria found in it are pathogenic bacteria. There are several different species and genuses. Heterotrophic bacteria, which is like a probiotic that produces metabolic substances that actually help to kill the pathogenic bacteria and prevent their growth, and then other contaminants which are not normally found in fish but can be found in it. And molds and yeast are some other types of microbes that grow on fish flesh. Seafood consumption and safety. So the highest risk of foodborne illness is associated with raw seafood consumption. And it is associated with things such as food poisoning, and that's your stomach upset that causes vomiting, diarrhea, headache. And then there are other types of foodborne illnesses, such as the ciguatera fish poisoning, which happens when you eat too much tropical fish, and that actually gives you a neurotoxin. And so you have neurological and cardiovascular issues with that. And then there's a gastrointestinal infection, which is an extreme form of food poisoning. If it progresses, it can become a gastrointestinal infection, which is just an infection of your intestines or stomach. Inspection, protection, and sale of seafood. So the Food and Drug Administration is in charge of regulating this, and they monitor everything from when the fish is caught through the shipping to the processing plant, the processing of the fish meat, and then also the packaging of the fish meat. And it all has to follow HACCP principles, which just outlines the safest way to produce sushi grade fish. And these laws are to protect consumers because they regulate the fish processing procedures, so that if it says sushi grade fish or just any other type of fish, you know that you're getting exactly what you expect because it will be prepared the same way. Materials. I had to make my own auger and my own petri dishes because of, obviously, we're working from home. So to make my auger, I used 1 packet of unflavored gelatin, 1 beef bouillon cube, 2 teaspoons of white sugar, and 1 cup of warm water, and then I boiled that. And once it came to a boil, I turned off the heat and I let it cool. And so while that was cooling, I had to take 20 plastic cups and I cut them off, so I just used the bottom. And I prepared those, wiped them with alcohol and sanitized them. And I also wiped down the toothpicks and sanitized them. And then I used the toothpick, and then I poured the auger into each of the 10 cup bottoms and then used the other 10 as lids. And so I used the toothpick to swab the salmon and then to smear it onto the auger. And then the Ziploc bag, I put the whole Petri plate into that to keep it sanitized as well. And so obviously I needed the fresh salmon fillet, frozen salmon fillet, and sushi grade salmon fillet. So my methods. I had to use sterile technique, the S smear onto the auger, and a 7-day incubation. Those are the main things. So like I said, sterile technique, that's where you have something sterile, my toothpick, and you touch the meat that you want to sample, so the type of fish meat that I was sampling, and then You go to the Petri plate, you lift the lid up just a little bit, just enough to get that toothpick in, and you smear it on and the S smear pattern onto the agar. And you don't press hard, you don't want to put any indents in the agar, you just want to wipe it gently. And then you close the lid of the Petri plate and throw away that toothpick because you cannot reuse it now. And that is the most sterile way to do that. And then I put it into the Ziploc bag and sealed the bag to ensure that no other bacteria got into it. And I left it for 7 days incubation. And I chose 7 days because fast bacteria will grow in about 2 to 3 days, and slower bacteria normally takes around 5 days. So just to be sure, I gave it 7 days incubation and room temperature, just sitting out on my kitchen counter. So the results, this is the control before incubation on the left, and then the control after incubation on the right. As you can see, there's not much change in these pictures. They actually are basically identical. The only change is that the right picture has this cloudiness in the agar, and that actually is due to my agar started to liquefy. I'm not sure exactly why, but upon researching it, it looks like the salt from the bouillon cube and then also not refrigerating it will make it soften over time. And here is the results from the fresh market salmon and the frozen salmon. So the fresh market salmon and frozen salmon, I did 3 trials of them, but I only included 1 picture for the presentation because they have the same information. The fresh market salmon clearly has a lot of growth. It has multiple colors. There's white bacteria, clear bacteria, green bacteria, and there's also that green patch of fuzzy mold. So there's a lot of growth on that. Most of the agar plate is covered with it. And the frozen salmon on the right has white bacteria growth and clear bacteria growth. And you can also see the cloudiness on both of these, especially on the right from the agar turning to liquid. Lastly, the sushi grade salmon results. So I included all 3 pictures so that you could see the wide variety of results. So the leftmost picture, there's not a single bacteria colony on it. There is absolutely no growth. And the right picture, there's almost no growth, but there's just that one little colony off on the right. And the middle picture has a significant amount of growth of white bacteria, which is an outlier. This is most likely due to some issue in sterile technique, but it could just have been because I was sampling different areas on the salmon for each smear, that maybe that spot that I had sampled was just a little bit more riddled with bacteria than the other spots. So the different bacteria present in each type of fish fillet. So you can see the control had 0, it didn't have any bacterial growth, and your fresh salmon had 2 to 3 types of bacteria, and that's where I went over the different colors earlier, where it had like white, clear, and green. And the frozen salmon had 1 to 2 different types because it had white or clear, and then the sushi grade salmon only had 1 type, which was the white type of bacteria. And so the average different types of bacteria growth on each sample, control has 0, obviously. The fresh salmon has 2.33 types. The frozen salmon has 1 type, and the sushi grade salmon has 0.67 types. So as a parent, the fresh salmon grew the most different types of bacteria, while the sushi grade salmon grew the least. And actually, it's interesting to note that the sushi grade salmon is 3 times less than the fresh salmon, and the frozen is about half as much bacteria growth as the fresh salmon. So the frozen salmon is actually still much safer to use than fresh salmon, but I'd still definitely suggest the sushi grade salmon. And so the types of microbes growth of each sample, so that is also including the mold and the yeast that was mentioned in the beginning. So not just the bacteria, but all different types of microbes. And so you have your control, which is 0. Your fresh salmon grew 4 because there's also the 3 that we mentioned earlier, and then that mold. The frozen salmon was still 2, and the sushi grade salmon was only 1. So you can see that it's pretty proportional, that it's the same result, that the frozen salmon had half as many as the fresh, and the sushi grade salmon had actually more than 3 times less than the fresh salmon in this instance. So discussions and conclusion. The sushi grade fish had the least amount of bacteria growth. The fresh market fish had the most amount of bacteria growth. And obviously, that leaves the frozen fish, which had the medium amount. It was in between. So sushi grade fish is safer to eat raw because there is less bacteria growth on it. And here's just my references. Thank you.